All right, I think we're live.
Awesome.
All right, today is going to be a little bit of a different live stream for you guys. I know you guys are more used to me talking about jumping and dunking and stuff like that, but today I have a special guest, Pranam Chatterjee. He was actually, one of his friends reached out to me from MIT to talk to him about CRISPR, and he works out of the MIT Media Lab, and he is doing research in nucleic acid nanotechnology, synthetic biology, and gene editing. So he's going to give you a brief summary of kind of the work he's doing with CRISPR and some of the other work out of his lab.
Yeah, hey guys, my name is Pranam. I'm a second-year graduate student at the Media Lab at MIT. I was actually an undergrad at MIT as well, where I studied computer science and molecular biology. But now the work that I primarily focus on is really applying new technologies to biological questions. So one of the technologies that I'm really excited about, and hopefully you guys will know about as well, is called CRISPR-Cas9. CRISPR is like a really cool new tool available for scientists to use, as well as engineers, doctors, to be able to precisely target specific stretches of DNA in your body. And this can be not only in humans, but it can also work in animals, in plants, and everything. And so really where this whole story starts is just a few years ago. It's a relatively new technology. And it was derived from bacteria, which is pretty cool because it's not something we invented from scratch as scientists. We found it, we discovered it using biology. And then what we realized was that, hey, this method of targeting a specific gene and then modifying it was applicable directly into other settings like humans and plants. And so what we've done really in the field in the last few years is try to make the CRISPR system, pretty much applicable to a lot of different problems. One of the cool things about CRISPR is that all you need to do gene editing is just an RNA molecule and an enzyme. It's just two parts. And previously, to do gene editing, what made it really difficult was that you needed a new protein every single time you wanted to go to a new gene. Just say you wanted to target one specific gene. You would have to go through an entire year of designing a protein just to go to that one specific gene. But now we're able to just say, hey, let me make a new RNA molecule, which takes about, you may not know, but it takes about like a day to do. It's very easy. Attach it to one enzyme, and it just goes to all these different locations. And so, and this is great for treating diseases. For example, one of my new CRISPR enzymes that I've developed is applicable to muscular dystrophy. So we're treating that disease right now in a preclinical setting. And then, we've licensed our technologies out to agriculture companies who use it to, modify plants or bacteria in many different ways. And, finally, people are like, what if we can change humans, right? Like, you can change them not just for disease settings, but just for cosmetic purposes, or to make you taller. Like I was talking to Michael, and I'm a pretty small guy, but I love basketball. I'd love to dunk one day, right? Like, and so maybe I'll figure out how to, use CRISPR to make myself taller. I don't know, but the point is that this technology is powerful, and we're trying to make it safe. And hopefully, in the next few years, we'll accomplish that goal.
I think that's a pretty good overall summary of the type of work you're doing. I think a question I've heard and I've thought of myself is how accurate is CRISPR right now, and how safe is it in its current state?
That's actually a great question, and it's what the main question going in when when CRISPR was discovered. How translatable is this? Because it's not a perfect system. What CRISPR has is kind of a phenomenon it has is called off-target effects. Basically, it hits targets that it's not supposed to. Right. Say you have a gene that looks like has specific DNA bases within it, but then you have another gene that looks almost identical, but maybe is one or two bases off, right? Mm-hmm. In those cases, the CRISPR system, original CRISPR system, would make that a mistake. Essentially, they'd go to both genes, and if you go to both genes, you could if that gene is essential, the one that you don't want to target, and then you accidentally modify it, could really create some bad side effects. And so what we've done, actually, I think we fixed this problem very well. So scientists at MIT mostly have actually come up with ways to look at the CRISPR protein itself. Remember, there's two parts, the RNA and the protein. We've looked at the protein itself and actually destabilized the protein at the wrong sites. So it won't cut. It won't modify at the wrong sites, but only at the correct sites. That was great, and it achieved a lot higher level of safety, like you said. But the next stage was what we did in the lab. Me and Noah, a grad student in the lab, we actually looked at the RNA molecule and said, hey, if we replace some of the RNA molecules with DNA, if we kind of just like switch some of those to DNA, what happens? DNA is what we call double-stranded, meaning it has two negatively charged backbones. DNA matched with DNA is two negative charges against two negative charges, and negative charges repel. So what happened was that when you introduced DNA, it would only bind, the CRISPR system would only go to sites that were perfectly matched. But if there was one wrong mismatch, right, like you were at the wrong site, it would not bind and cut. So we've really tuned the safety and the specificity of this system so that now we only go to the correct sites and not cut the wrong ones. And so I think we've really solved that problem. And again, this is work that is going currently into clinical trials for genetic diseases.
Okay, so you said, so right now most of the work is just happening in more of a laboratory setting and just now moving into
exactly. Yeah, because I think the system has only been developed for 2 years or so, like 2-3 years, and so we are now actually the first human trials begin this year. So which is really cool. I mean, this is the fastest thing. Usually drugs take 20, 30 years to get from the lab to usage. For us, it has taken 3 years, and we're almost there. And in fact, people like other countries like China, where there's little less regulations, they're already there. You're already at the place where it's already being used in humans and it's working, which is really cool.
Okay. If you want to talk a little bit about Cas9, the actual protein, and maybe even how you use the dead version of Cas9.
Yes, it's actually an excellent thing. The original Cas9, which is the CRISPR protein, its main ability is to cut a specific gene, right? So you take the RNA, attach the Cas9, it goes to a specific piece of DNA, and then it makes a cut. And when it makes a cut, it induces one of two things. Either it creates a mutation in the gene, which essentially silences the gene or inactivates the gene. It can't do what it's supposed to do. Or you can, at that cut site, you can introduce a new piece of DNA in its place. So that's a method of either gene silencing or gene insertion, right? That's really powerful. That was the original system. Then we realized that there was two parts of the Cas9 protein that if we change the amino acid, essentially the part of the building block of the protein, we just change 2 of the 1,000 amino acids, we could actually deactivate it, as you said, a deactivated Cas9, a D-Cas9 we call it. And so what we were able to do now think about it, you don't have cut, you can essentially just take things to places in the genome. What if you want to add some chemical modification? Just attach that modifier to Cas9, to the deactivated Cas9, go to the site, and it'll modify the DNA. A cool thing that came out last few months ago is a thing called a base editor. So no longer do we want to just cut and then messiness happens and we remove the gene. Now we can go to a specific site with a deactivated Cas9 and then selectively convert a base, let's say A, right? A, C, G, and T, convert an A to a G or a C to a T, right? And so now we can do very clean gene editing on a single basis, because most diseases and most, any kind of genetic problem that people may have are actually caused by single mutations, single one base mutations. Right. You don't want to mess up an entire gene just to correct one mutation, right? This is a way that we've repurposed the deactivated version of Cas9 to actually do this single base modification, which is really cool.
So with doing single base modification, would that lower the risk of off-site mutations, or is there still that risk with off-site mutations, even with single base pair changes?
Remember, the CRISPR system itself has this off-target propensity, right? It has that, and so the thing that does the base changing is just attached to the CRISPR system, right? To the activated CRISPR system. So you would still the off-target off-target phenomenon still holds in the base editor, but with these new, like I said before, these new off-target mediating, correcting technologies that we've developed in the lab, just attach that to the base editor architecture, and you reduce off-target effects. So, yeah.
So something else I've kind of read about and thought about is how does our immune system kind of either reject or accept CRISPR modifications?
That's a great question, and that actually came up out a few months ago. And I remember reading this article. I was like, "Oh, there's immunogenicity to the Cas9 protein," and I was like, "Okay, like I'm not surprised. Why? Why am I not surprised? Because remember, CRISPR comes from bacteria." And the current CRISPR system that everyone uses comes from a very specific bacteria. It's called Streptococcus pyogenes. Remember, you can think about it a little bit. It sounds like strep, right? You've probably had strep throat. That's caused by Streptococcus pyogenes, essentially. So we got that Cas9 from that bacteria, and so that bacteria infects humans, right? And so we already mount an immune response to this bacteria. So it's not surprising that a Cas9 from a bacteria that infects humans also has elicits an immune response. So it's not surprising. They did another Cas9 from Staphylococcus aureus. You probably heard of that too, staph, right? Like staph infections, right? And it also infects humans. These two are the only two Cas9s that they actually studied in the study. And so no one in the CRISPR community was very surprised. So the way to fix this immunogenicity problem, either is you find a Cas9 that is from a bacteria that is not infecting humans, or you can engineer the current Cas9 enzyme to not be immunogenic, have an immunogenic effect. You can essentially just like modify the amino acids. What I've done, my Cas9 enzyme is actually from a species that doesn't infect humans, and so my Cas9 enzyme, which we're about to publish soon, actually doesn't have immunogenicity in human cells, which will be a very nice, thus making a pretty nice therapeutic. But you're right, it's definitely a concern. It actually tanked a lot of CRISPR stocks when it happened. So, but that's because people I guess investors don't terribly understand the system very well.
Right. Lately, the stock has been going up a lot, actually.
Exactly. Hopefully, you have some stock in it. Well, I mean, everyone kind of does. I have licensed some of my technologies, or we're licensing some of our technologies out to certain CRISPR companies. Right now. Okay. Yeah, so there'll be.
You talked about the base editing.
Yep.
Is Beam Therapeutics? Is that one of the companies?
Yep, it just started. It was big news, and it was started by the guys who actually designed the base editor. And so we're working with people at Beam to put our CRISPR enzymes into their base editing architecture. Actually, today, just like an hour ago, I did some base editing experiments with my new CRISPR enzymes. And pretty much the point of my CRISPR enzymes is that in the CRISPR system, this is actually one of the main, I told you off-target effects is one of the big drawbacks. The second drawback is that we actually can't go to every gene in the body. Why I'm saying that is that there's this specific restriction called the PAM sequence. It's like this little sequence that has to be beside the sequence that you're trying to target. That has to be there for the CRISPR enzyme to be able to do its thing. And so the current restriction is that you need 2 Gs beside the target site. So if you think about it, 2 Gs, that's 1 over 4 to the second, right? Because there's 4 bases. You can only go to 1/16 of the entire genome with the current CRISPR enzyme.
And right now it's about 20,000 known? Is that right?
Yeah, but there are tons of CRISPR enzymes out there. Is that what you were saying?
20,000 known genes in the human genome?
Yeah, 20,000
protein encoding.
Protein encoding genes, which are the ones that you want to target, essentially, right? But you could also target other regions as well. Mm-hmm. But if you just want to, silence a gene, no big deal, right? Like you can definitely find a GG somewhere in your gene, look beside it, and then cut there, right? Or modify there. Not a big deal. But what if you want to edit a specific base, right? The mutation is right there. You better have a GG right beside that. Otherwise, you will have no ability to fix it. And so the way to do this is to minimize that PAM sequence, essentially, right? Essentially, get it so that you don't have that sequence restriction. So this new Cas9 enzyme that I was talking about, what makes it so special is that it only needs a single G. So now you can target 1/4 of the entire genome with my new CRISPR enzyme that we're doing. And then we're also working on another platform to work for 1A, to only need 1A. And so now, remember, DNA is double-stranded. And so if you can target a G, you can also target a C. You just have to go at the other side of the gene. So if you want to target a, what do you call an A, you can also target a T. So if you just have a CRISPR enzyme that has a single G requirement and a CRISPR enzyme that has a single A requirement, you cover the entire genome. And so you can go to any place. And so those are the technologies that I've developed.
Okay.
Yeah, and that's very useful for base editor for that reason.
So I guess another question: how are you using computer science there in bioinformatics to kind of make the whole process with CRISPR faster and easier? Because it seems like it's just becoming exponential in terms of how fast we can sequence the genomes, and I'm just wondering how you're using CRISPR, applying CRISPR to or with bioinformatics.
Exactly. It's actually, I mean, remember I told you that CRISPR comes from bacteria, and guess how many bacteria there are out there? There's like, I mean, millions, right? Like so many bacterial species, and they grow so fast, and they have so many different properties. The thing is, when you want to find a new CRISPR enzyme, right, you have to find a way to search through, all of the, like, there's at least 3,000-4,000 known Cas9s, but there's infinite numbers of bacteria that we have not gone after. And so we use big data. I use machine learning to actually search through the current sets of CRISPR enzymes to find the CRISPR enzyme that I want, right? Just say I want something that has a very short PAM sequence, right? Like then I would use machine learning to actually search through and learn what is the sequence, what is the optimal sequence that you'd want of your CRISPR enzyme so that it doesn't need a long PAM sequence. And so that's like one application. Another application is if you wanted to find out what is the most effective RNA sequences. Like just say you have like 10 choices of where you could target in the gene, which one is the best one, right? And so we use machine learning to decide, based on the sequence of the guide of the RNA molecule, can you tell me what is the predicted efficacy of the CRISPR system? So we can predict how well a CRISPR system will work at a specific DNA sequence.
I'm sure with machine learning, that just becomes exponentially more accurate.
It does, because guess what? You have like so many. I think in our data sets are huge. They're like 100, 200 to million different sequences that are targetable, right? And so you want to be able to push through all of that data through a model, and really the best models for that are machine learning.
Okay.
Yeah, that use machine learning, and so I think we're getting really good at like predicting how well CRISPR will work before we even do any experiments.
So is there any potential in using a virus instead of a bacteria to kind of minimize that immune response that you talked about earlier?
Yeah, that's a good question. I mean, the people are looking at methods of delivering gene editing systems to the body, right? One way is actually using a virus. Like viruses that are not lysogenic, so they will not negatively affect the human being it goes into or whoever it goes into. What it'll do is it'll just integrate whatever you want to change into the genome. So the idea of AAV, we call it adeno-associated viruses, are actually pretty popular gene therapy platform. Unfortunately, you can't go to a lot of places with it. And so you don't have the ability to go to any gene and modify it. You can go to some of them, and actually viruses work really well to modify certain spots in the genome. But the CRISPR system, what makes it so special and beautiful is that you can go to almost any site, right? Almost any gene.
Does that have to do with bacteria being a living organism and a virus not? Or
well, remember the Cas9 and CRISPR enzyme itself doesn't use the bacteria is completely gone by that time, right? We've isolated the protein from the bacteria. So what we do to deliver the CRISPR system is we actually package it within a nanoparticle, like essentially like a synthetic thing that is not a synthetic, like a capsule that is not dangerous to human beings and can actually be endocytosed, like pretty much taken within the cell. And so that's how actually the best method of delivery of gene editing systems. Viruses are another method, but we still have to figure out which is the best platform going forward.
Okay, so I guess just to get into the nanotechnology side of things. So is nanotechnology just basically making precision or medicine more precise in terms of delivering a treatment? Is that
That's exactly what nanotechnology is, right? Like at this point of of course, nanotechnology is going to go beyond like living or like organic molecules, which is what they are right now, which is like, you know, biological molecules that we're using to assemble into nanostructures that can deliver things. I mean, obviously, people in my lab are actually working on like inorganic methods, like metals and small nanorobots that are made of like non-carbon based material to deliver things, not in the human body, but in other applications. However, you're correct. Right now, the big challenge is that, well, how do we formulate these small little nanorobots that can carry things to different cells, different parts of the human body? Because guess what? You know, you may want, just say you want to target a cancer cell, right? Like, and you don't want to, you know, go after a cell that is fine, right? You only want to edit the genes of a cancer cell to make it maybe non-cancerous. Then you want a method to only target cancer cells but not go after. The way we do that is we encapsulate whatever gene editing system in a nanoparticle, and then on top of the nanoparticle, we attach antibodies that are specific for a specific cell type. For example, a cancer cell expresses, you know, a specific receptor that only cancer cells have, and so we're going to attach an antibody to that receptor, put it on top of the nanoparticle, and deliver it. And so then it just goes to the cancer cell, and then the CRISPR system can go and modify that cell. And so that's the kind of the level of nanotechnology that we're on right now.
Okay, and then before the live stream started, we were talking a little bit about immunotherapy. Would you want to get into kind of some of the work you've done with immunotherapy and how that's being used in medicine?
Exactly. It's actually, you know, if it wasn't for CRISPR, immunotherapy would be pretty much the biggest, I would say, biomedical discovery of the decade, or maybe the century, one of at least the last few decades. CRISPR is obviously like insanely amazing for so many different applications, but right now, most of the cancer therapies are immunotherapies, or the safer treatments are immunotherapies. The reason is the immune system allows you to specifically target, like the immune system is very specific to different types of cells. For example, your T cells that actually mediate the fight against cancer, for example. When it's in, what we've discovered was in the tumor microenvironment, basically where the cancer is, the cancer cells are smart. They developed a way to actually attach to the immune cells and turn off their metabolic abilities, essentially really like give them no energy to work with. And so these cells can no longer fight the tumor. And so the tumor persists and it grows and it metastasizes. And so what I worked on was this pretty much my entire undergrad. For 4 years in undergrad, I worked on this one molecule called PD-1. And it was a molecule that was expressed on the surface of your T cells, of your immune cells. And so cancer cells developed this thing called PD ligand 1, essentially ligates, like attaches to PD-1. And when it attached to PD-1, what I discovered, one of my first paper I ever published as an undergrad, was showing that when that happens, when that attachment happens, it essentially allows the T cell's metabolism to go from using glucose, like sugar, right, regular sugar, to actually using what we call lipid, essentially using a lipid fatty acid oxidation, like using a much less efficient energy source. And so the cells would just like kind of go sleep, essentially.
Would that be different than using a ketone, or is it similar to that?
It's similar. If ketones are very processable by cells, so that cells could survive on it. These cells don't die, right? You see, like they don't die. They just cannot survive on that alone as their energy source. The body needs to be able to. Your cells in your body need to be able to convert its resources into sugar, simple sugars like glucose. And if you can't do that, then they cannot remain active cells. And so your T cells shut off, and you're like, shoot, like I can't clear this cancer anymore. And so what we did, I actually interned at Pfizer, where I was working in academic labs, and one summer interned at Pfizer, where I built an antibody that would block the PD-1 receptor from being ligated by PD-L1, and so that would keep the T cells active, and then allow them to keep fighting the tumor. Unfortunately, my antibody did not pass phase 1 because there was some toxicity, but Bristol-Myers, I was at Pfizer, and Bristol-Myers Squibb, another pharmaceutical, came out with the exact same thing, just a more improved version, and now it's the standard of therapy for melanomas and blood cancers. So Yeah, so it's a very cool technology. This is all immunotherapy has unleashed this like power of just targeted cancer treatment.
So I was reading a little bit about immunotherapy recently, and something I came across that you might be able to answer is why does immunotherapy work for some people and not for other people?
Yeah, and that's I have a friend whose brother had a melanoma and did not and so there he originally enlisted for a clinical trial, and so he unfortunately he did not survive that clinical trial because he did not express PD1 on his cells. So PD1 is bad if you don't want your cells to be, turned off. But if you don't have them in the first place, then the cancer cell is using an alternate mechanism that is not suppressing the immune system. And so if you don't express, that's why all these pharmaceuticals will put out advertisements saying if you'll first be screened to see if your cells express the immune receptors, and if they do express the immune receptors, then you're eligible for an immunotherapy. But again, we're all different, right? Like your immune system is probably different than mine, and so not everyone will be able to respond to these therapies.
We were also taught in one of my biotechnology classes. I don't know how true this is now, but we were basically told that immunotherapies, gene therapies, and other maybe uses with CRISPR, those aren't the go-to treatments right now. And is that just because of the accuracy or the way they're affecting off-site mutations?
Yeah, you're right. I mean, that's actually really interesting, and we understand first that these are young technologies, right? Like, and for the most part, we actually, for immunotherapy, let me talk a little first, is that we don't have an immunotherapy for every type of tumor, for example, or cancer cell. And so we don't know, we cannot always, attack tumors that may be metastasizing in a different part of the body because the immune cells that we're trying to target are not there. So that's one thing. For CRISPR, obviously, it's because gene therapy is great in practice if you want to make a single change. But if you want to make multiple changes, it's a little bit more difficult. We don't.
Right now, they're basically what you said, like single nucleotide polymorphism, so like sickle cell anemia. And you said you're doing work with muscular dystrophy.
Correct. Yes, we're trying. And muscular dystrophy is a little easier because you can go. It's actually one big region that you need to cut out, essentially. Which is CRISPR does really well, which you can just cut out big parts of the genes. And so we're almost to the point where we can do all the different genetic fixes. As I said, base editing, inserting genes or removing genes. And so we hope that these technologies will all be safe enough in the next 2-3 years. I expect that all genetic diseases will be cured in the next 5-10 years because of CRISPR's efficacy as well as their pretty relative level of safety going forward.
Even beyond targeting single regions?
Exactly. I mean, beyond single regions is, super powerful, and we're able to do that. But what we're working on now, the field is working on now, is what we call something called multiplexing, basically going to multiple sites at once. And you can do that if you have different CRISPR enzymes all being delivered together. So in the end, it becomes a delivery problem. How do you put all of this in the body such that it works well with one try?
Okay.
Yeah.
So I guess this is like a more general question, but is it true that the estimate right now for how many base pairs we have in our entire genome is around 3 billion?
Yeah.
3 billion base pairs?
That's about the correct estimate. Yeah.
So are we using machine learning to try and basically read out that entire, I guess, booklet, you can say, of base pairs?
Well, I don't think in this case machine learning may not be the best strategy. I think we are good at sequencing genomes, right? You see like all these companies that are now out there just to sequence genomes, right?
I think it was just recently there was the first one completed in under 24 hours, I think. It was
yes, exactly. But remember, machine learning is only as good as the data sets you provided, right? Like, and so we have good amounts of data sets, but if you're going to create like synthetic genomes, right? Like if you're just going to like read out what is the predicted best genome, or like that would not have any like health effects or anything, machine learning would be a great approach. But we're still to the point where we're sequencing a lot, right? We're still gaining all this data, and so we need multiple sets of genomes, like everyone's genomes, to get to the point where we can understand, okay, here are all the different variants of this specific gene that everyone has. Which one people are healthy, which people are not healthy. we are almost at a level where we can predict that on a medical level using machine learning. So sequencing technologies are independent of machine learning, but sequencing results will get you data that you can feed into your machine learning models. Right. That can give you cool information about health prognoses.
And do you think this, or I mean, it might be currently, but do you think this is going to be used to, like all this data is going to be used to actually formulate new prescription drugs and stuff like that?
That's a great question. And like the reason I say that is that the other half of my research group is focused on using machine learning to actually generate new drugs. So not on not looking at human genomic sequences, nothing like that. What they're doing is that they're learning the interaction. So machine learning doesn't have to just be on like a sequence level. It can be anything that you can represent in a data format, right? Like just say you want to represent a chemical molecule as a piece of data. You can like convert it into numbers that will represent what that molecule is. And so what they've done is use that information, like the structure of specific proteins, and say, can I design a chemistry like using machine learning that'll bind to all of these different proteins? And so now you can create drugs to sites that are not that we previously weren't able to target using machine learning. Have they been successful at that? Not really. But there are legitimately like 4 or 5 companies that have raised, $70, $80 million in initial seed funding just to answer these questions.
Yeah, I heard 23andMe is possibly one of those companies who's going to start their own almost like pharmaceutical company to use the data they have to start.
Yeah, they have access to so much data like that they like privately hold, and so they would have a huge advantage in this field.
So what do you think is, in terms of like holding that information as confidential, do you think they're going to run into any problems with using people's genomic data to try and formulate medicines?
I do believe that they have some agreement with the people who submit their samples, whether they can use that data anonymously for machine learning or whatever data visualization purposes. So they probably sign a contract, but yeah, I mean, we are getting to the point where personal data is freely available. We saw with Facebook, right? It's a good example, but this is even more personal if you can imagine that, right? Like it's your actual health and your own genome, right? It's kind of dangerous, and so yeah, and I can totally see lawsuits and legal ramifications arising as a result of like this huge data revolution that we're undergoing, both in medicine as well as in just like social media.
So do you think this is eventually going to end up in like truly personalized medicine, or you think it's just going to become more precise and not so much personalized, maybe?
Yeah, I understand. I see what you're saying, and definitely precision medicine is like personalized medicine is what's going to be the standard of treatment in the future. CRISPR is great because even if the gene is different, for just say we have the same disease, just say 2 people have the same disease, but their genes are different, right? They have some differences that are not causing that mutation, not causing that disease, but you want to target that gene. So now CRISPR, it's easy, right? You can still go to the different genes of the different people without any difficulty. So that's great on a genetic level. We're almost 100% capable of doing that. For like immunotherapies or cancers, we are really good at understanding the specific cancer profile of patients, right? What type of immune level do they have? What is their where is the cancer metastasizing to? And using that information coupled with machine learning, because machine learning can tell us what treatments are effective, right? From what
people's data, right?
Exactly. And remember, machine learning is not just like, I saw this, and therefore if I see it again, I will know what to do. That's a standard computer science. It's like
more like comparing across.
Exactly. So even it's the idea of generalizing, right? Like, so I learn that if you have these like blood count levels and these phenotypes, this is the outcome, right? But if I have a person who's completely different, right, like has all these different things, I still can learn the parameters from the model that I have to apply it to some new problem, a new patient, right? And so that's where it becomes super personalized, right? you can still use the information of all the previous patients to be able to treat a patient that is completely different.
So how close do you think we are to walking to a doctor and them and it being kind of a regular process to ask somebody to get their genome sequence and then prescribe off of that?
So it's not actually as clean as that, in the sense that we used to think so biology has undergone three big revolutions. It underwent the sequencing revolution in the early 2000s, the synthesis revolution in the late 2000s, and then in this decade, the editing revolution, right? The CRISPR and Cas9. So we thought I was too young, but when sequencing came out, everyone thought that, we sequence a genome, that will give us they'll hold all the answers to. It actually didn't. And that in itself is not enough unless it's like an obvious genetic disease. Usually genomics is it's not an end all be all thing, because we have things like epigenetics, which are where chemical modifications of DNA. We have
proteomes,
right? That we have to undergo. These are not as simple as sequencing your DNA and then seeing what is in there. It's a much more complicated question. But having the ability to sequence a genome very quickly and cheaply adds to the predictive ability, I think, of the machine learning models, not necessarily doctors. I personally think doctors will be obsolete. At least outpatient doctors will be very obsolete, because they're legitimately looking at numbers and making conclusions of numbers, which machine learning models or any computational models do perfectly, accurately, and deterministically, right? And so
would you say better and faster, maybe?
I think better and faster, and people are like, we need doctors for personal human to human interventions. I'm like, personally, I think nurses do that much better, but maybe it's a controversial opinion. I think surgeons will last a little longer, but obviously robotic surgery is coming. Medicine is the perfect target for automation as well as precision medicine, right? CRISPR and AI are the two greatest technologies of our time, I would say, and they're both coming together to solve all of these biological questions.
So do you think one day in the future, this is actually for one of our projects in biotechnology, we had to come up with some hypothetical technology that can possibly, become useful to society, I guess, one day. And I kind of proposed an internal genomic device that is able to track changes to our gene expression depending on, what food you ate, if you exercised. Do you think one day we can get to something like that with the way machine learning is going and technology in general?
I think it's very cool, and you would be surprised. Well, just 2 days ago, there was a paper published out of MIT that essentially did this. They have chips that are embedded in the human body that detects levels of mRNA. mRNA is the byproduct of DNA, is the intermediate product of DNA and proteins, right? And so you need mRNA. So they can track changes of mRNA using an implantable chip, which is essentially what you said.
And we actually got the idea from was reading an article. I forgot what the website was, but it was a study published from, it was either Harvard or MIT, talking about camera technology.
The camera is interesting because camera actually uses base editing. It's a method of tracking changes in your DNA. Actually, the other grad student who's upstairs right now, he's really good at that. This is what he did for his master's thesis. Track. He's actually coming in right now. He may say something, but he essentially tracking changes in the genome over time and being able to read it out in real time. Yeah, and so that kind of technology is very powerful. Just on a preventative medicine type level. Who knows where it'll go? Who knows how it'll go in a real time setting? So I think it's I think it's really cool that you thought of that already.
Yeah, because I think one of the uses for it was actually being able to track how long somebody's been on a certain antibiotic based on changes to cell signaling and changes like that.
Yeah, no, I think you're absolutely right. There's obviously a lot of once you get to the genomic level, right? There's a lot of things you can track that are independent of the reason is your genomic state is very indicative of a lot of different factors that you could essentially learn, right? You could essentially understand what are the different parameters in play just by knowing exactly the readout of a system like you described.
Mhm.
So yeah, I think it's very cool. That would be very cool.
So I don't know how much of your interest is in nutrition or nutrigenomics, but how much or how long do you think it'll take before we can actually track changes to someone's genome just based on maybe like the foods they eat and
It's a good question. I don't know. Like, it's hmm. The first step would be something like this, right? Like you would have to have a really nice controlled experiments where, based on like just nutritional intake, can you have a real-time tracker of mRNA levels, protein expression, all these maybe epigenetic modification, something that just gives you an idea of what is the global effects.
Yeah, that seems to be one of the biggest issues is you have to take into account, the whole picture. It's hard to just say, well, you ate this food and that's exactly what caused.
Maybe the hardest controlled experiment I can think of, right? Like it's a systems biology level thing, and really maybe the first way to do it is to do a computational model of it. Like, can you without doing it perfectly controlled, can I use, because there will be so many variables, right? Like you can monitor all these different changes in the body with different food intakes. Can you take all of that information and then maybe reduce the dimensionality of that data so that it's not like a 100D problem where there's 100 parameters? Maybe you can reduce it to 3 important things that are critical for and then you can isolate those things and go forward with that as predictive markers. So yeah, that's as you can say that there's a long process. I think that's like going to be one of the most sophisticated biotechnological solutions we would ever get.
Because I think it'd be interesting to one day have like truly personalized nutrition where we can base somebody's diet based off of their genomic makeup. And
yeah, I mean, it's already personalized, right? Like to the point where people kind of generally understand what is good and bad for them, and based on more easily measurable things like body mass, BMI, and all these external phenotypes, right? Like we can weight and stuff. You can understand what is good for your body, but it's not personal until you get to the probably the genomic level, right? Yeah.
And who knows? Maybe there'll be something beyond that soon that we'll find out.
That would be very cool. I think that would be technology that people would instantly or doctors would essentially. The medical field will essentially be using very quickly if it came out, but we'll see. It's not clear yet whether that's going to be happening in the near future.
Yeah, and before the live stream, we were talking about one of the studies that you've done with ID2 expression.
Yeah.
And glioblastoma survival based on glucose deprivation and other treatments like that. Would you want to talk about that a little bit?
Yeah, I mean, it's been a while, and I was telling you beforehand that all the research that is like what is not the ugly duckling, but just like the an out of place. I'd worked in this lab for a few years, and I was in between the work that I'm doing now and the work I did in immunotherapy. But ID2 is really cool because it is specifically expressed in glioblastomas, and so what we were able to see is that when ID2 is knocked out,
those are brain tumors, right?
Yeah, these are glioblastomas are essentially brain tumors. When ID2 is knocked out, we see a shift in these cells' metabolic profile, right? Just like I showed you, T cells, when PD-1 was ligated, altered their metabolic profile. ID2 is one of those governing genes in the metabolism of cancer cells. Have we designed a therapy for it? Not really, but you can imagine that being another form of targeted therapy, right? Like, can we go directly to these cells and just inhibit this? And you can do that, and now you see a very strong connection to everything, right? You're going from like targeted immunotherapy, now go to take gene editing, like CRISPR, and apply it to modifying the ID2 gene in glioblastoma, targeted with a nanoparticle, a nanotechnology, go to those cells and then edit out that gene. And maybe you can reduce the ability of those cancer cells to survive. And so it's like, it all comes full circle, I think.
So what I found interesting in that study was the glucose deprivation side of it. So would you think, or would you go as far as to say that this might be too much of a stretch, but saying that a high-fat diet can possibly lower your risk of certain cancers because of that?
It's not clear. I mean, again, this is something that is and this is, it's hard. I would be afraid of making these overgeneralizations because it's the point is that in the end, your body processes pretty much everything you eat, right? And in the end, the cells use glucose as its main energy source, but that glucose is not derived simply directly from the foods you eat, right? It's derived from the the breakdown of those nutrients into the smaller building blocks, and so it's difficult. Would I say that is a plausible hypothesis? Yes. But to actually test it, we would need to do more molecular-based studies. Can you go ahead?
I was gonna say that's why I think doing molecular research and research that you're doing is so important, because I feel like there's been so many different diets thrown out there with like ketogenic or fasting or higher carb, lower fat. It's just there's so many different diets out there that people might think is affecting them in one way, but it might be a broad generalization to make.
This is a huge dimensionality reduction problem, right? Like how do you take all of the different bodily effects of the food you eat and say that this is directly causing this, right? Like to happen. You can't really do that now, but it's a very ambitious thing to think about. That doesn't mean that you can't obviously go on specific diets and it'll help you, but it means that really what I say as a scientist is, to be honest, you need to really run controlled experiments before you can make very strong conclusions like that.
Right. It's just hard to say that this one diet will explicitly affect you in this way.
Exactly. It's very. I'm always scared of these generalizables. I mean, molecular biology is so constrained, right? You're working legitimately at the molecular level that you can really control things. Like, I'm just putting a protein in. Let's see what happens. I'm putting a protein in. But like when you put food in, you're putting all this stuff in, and you're like,
so many different chemicals are going in. Yeah.
Chemicals, and how are they being all processed? All the organs, and it becomes a very difficult question. There are methods you can think about how to study this, though, and I'm sure nutritional scientists are using strategies to deconvolute this problem.
Right. But it almost seems like it boils back down to molecular biology and computer science, because
computer science is the user of this data that molecular biology and genomics and produces, right? So and then it's a cycle. It feeds back into the cycle. I use machine learning to discover new CRISPR proteins. I'll go back into the body. That'll create more data for me to learn to use machine learning, and the circle continues.
Right. Well, I think that's about everything I wanted to talk about. So thank you for taking the time out to do this. I'm definitely going to watch this again, because so much information coming up.
Awesome. Yeah. No. I hope you enjoy it, and I hope people start to realize that the CRISPR system is, and any of these technologies, they're coming, and in the next few years you'll see it on, and it's good to understand it, because if we don't have people understanding the technologies, they're either afraid of it or are more reluctant for the entire society to adopt them, right? And I think a goal of mine is to make sure that people understand how it works, so that when it comes, they're not left in the dark.
Right. Yeah, I agree. I think it's important that people are aware of the type of work that you're doing and other scientists are doing with this technology. So
yeah, thank you for saying that. Thanks so much for having me on.
Oh yeah, thank you for taking the time again.
Appreciate it very much.
All right. Bye, Pranam.
See you, Michael.
See you later.